In the arid stretches of the Middle East, a lack of fresh water leaves populations relying almost entirely on the ocean for survival. This scarcity isn't limited to deserts. Even lush tropical island nations face extreme freshwater deficits, despite being surrounded by seawater. To meet this demand, the modern world relies on industrial desalination, producing billions of gallons of drinkable water every single day. The primary technology used is reverse osmosis, a process that physically forces seawater through fine membranes to filter out the salt. This mechanical extraction requires massive capital expenditure. To maintain the intense pressure required to push water through those membranes, these plants consume enormous amounts of fossil fuel energy. Reverse osmosis solves an immediate thirst, but it is a brute force mechanism. It trades a water crisis for an energy and environmental one. The extraction process creates a problematic byproduct. For every gallon of fresh water produced, a highly concentrated super saline brine is left behind. Because of the scale of production, industrial facilities often dispose of this waste by continuously dumping it directly back into the marine ecosystem. This brine is hyperdense. It sinks to the ocean floor, suffocating benthic aquatic life and artificially spiking local salinity levels. By introducing this concentrated discharge, industrial desalination disrupts the marine balance, essentially poisoning the sea in order to drink from it. Long before industrial plants, the Earth managed its own mineral levels through a continuous natural cycle. The cycle begins on land. Rivers physically break minerals away from rocks, carrying them downstream and into the sea, which creates the ocean's baseline salinity. That saline water eventually moves into underground aquifers, where it is filtered through soil layers and returned to the landmass. The biological engine powering this filtration consists of microscopic cyanobacteria, like Anabaena tortuosa, which thrive in high salt environments. These microbes use the salt to survive. They actively trap sodium ions within their own cellular structures, neutralizing the salinity. This microbial desalination functions without producing a concentrated waste stream, but the natural speed of the process is too slow to meet the demands of modern human consumption. Countonomics technology was developed as a biological catalyst to accelerate this ancient microbial process without relying on heavy machinery. The application is simple. A highly diluted biological concentrate is released directly into a body of brackish water. When this solution enters the water, it acts as a molecular trigger, supplying free electrons and shifting the water to a negative oxidation reduction potential. The negative charge of these free electrons physically breaks the bond between the sodium and chloride in the salt water. The isolated chloride then oxidizes into chlorine gas and evaporates out of the water. The remaining sodium atoms are now available to be eagerly consumed by the native microbial biome, which was triggered into action by the electron charge. This influx of energy transforms a passive pond into an active, high-speed biological desalination engine. Unlike the industrial plant, this biological method requires no massive energy grid and introduces no invasive species to the environment. Because the salt is either evaporated or integrated into the microbial food chain, the process yields zero toxic brine. Scaling this approach could eliminate the threat of brine discharge for coastal and island nations by working within nature's existing limits. Sustainability is not achieved through engineering that fights the environment. It requires aligning our technology with the Earth's oldest microscopic mechanisms to restore the water cycle.